All right. Thank you everyone for joining for our last masterclass session. I'm going to go ahead and get started. So this is our last GeoMx data analysis masterclass session. Today we're going to be focusing on spatial differential expression. If you've been here with us through the previous sessions, thank you for being here. If you haven't, we do have recordings of all these sessions. You can find them on our Rosalind Academy, or you can reach out and ask us for the recordings and we can send you the link. But thank you for being here today. So our previous sessions started on September 12th, and essentially these are a step-by-step basis on how you should, there's no, required step-by-step and how you should look at your data, but this is recommended step-by-step and how you would approach your spatial GeoMx WTA data, and some tips and tricks and how you would be taking a deep dive into your data quality and analysis for each of these steps. And so they started with QC statistics, AOI and gene removal. The last session was normalization selection and different options for normalization, how to take those into effect. And really it's a step-by-step how to take each of these into effect in your model, in your data, and unique to your type of situation. And then going into that next step, because they really do take effect and build and talk onto each other. And so finally, we're in this last step where now that you're really confident in your data and the data that you have and how it's been normalized, now we want to finish this off with our spatial differential expression and learn how this is really important and still a key step in how you want to look at your spatial data. And so what I hope you've learned throughout this masterclass, or if you haven't watched all these series yet and you plan to go back and watch all of them, I really hope that throughout this series you've learned how to solidify the value and synergy of pairing Rosalind and GeoMx at your institution or within your lab, gain a deep understanding of spatial analysis principles and algorithms and how Rosalind can simplify your spatial analysis experience, increase your confidence to accurately process and interpret your spatial data and results, and open new avenues for insight that you may have missed. So again, some of this is going to be a bit repetitive from these other sessions, but for those of you who may be new, Rosalind has built a beautiful new pipeline to analyze GeoMx data for WTA, or whole transcriptome analysis. We've taken a deep dive into each of these steps themselves for data, as well as these steps that Rosalind has developed for each of these steps. And so we've gone through each of these items and really how Rosalind can elevate each of these items for your analysis. And today we're going to be finally focusing on this last section for guided differential expression, how this is still a really key factor in your data and how you should really be thinking about this and processing this in your data analysis, and how Rosalind is really taking this to the next step and again making this the most powerful and collaborative solution for GeoMx data, WTA data analysis on the market today. And the goal of this, again, from each of these steps is really creating this end-to-end solution with Rosalind and GeoMx together to allow you rapid discovery and deep insight, beginning all the way from the bench as you're staining your tissue, really focusing on that project design and region of interest selection, to sequencing and collecting your data, inputting that raw data into Rosalind, and again, getting that very rapid discovery, feeling very confident in that data that you're getting very quickly, bringing you all the way into your very next project as fast as possible and as confident as possible. And so a little bit about me. I have been leading this masterclass. So again, I apologize if you've heard this slide 4 times, but for those of you who are new, I have a PhD in biomedical sciences from UC Riverside. My graduate studies were in translational ischemic stroke research with neuregulin 1 treatment. I have multiple publications in spatial biology and data analysis, and I was previously a nanostring field application scientist. So my expertise really does lie in spatial biology, immunohistochemistry, and neuropathology. And so I'm really excited, to have helped build this pipeline with Rosalind and bringing this masterclass to you all with tips and tricks from my own research, as well as working with countless researchers in various different fields as my work as a FAS at NanoString. And so I'm really excited to be helping you and sharing this knowledge across these different steps with GeoMx WTA analysis. And today we're finally talking about differential expression. So in a very vague, generic description, why is it important to perform differential expression? So differential expression helps us determine what genes or proteins or factors or pathways can demonstrate an observed difference or response between conditions or a model. But in a deeper level, what are some examples of why someone may want to perform differential expression? This can help us understand biological differences between conditions, identify key biomarkers, deepen an understanding of a biological model or disease or treatment, and even identify an underlying mechanism of action. More importantly, this helps statistically determine if sets of data are significantly different from each other. So this helps increase that confidence for discovery, publish data, and perform follow-up experiments or submit for peer review. So why do we want to think about this in terms of spatial differentiation and how we perform that? So I like to compare how we approach this with Rosalind and how I'm going to show you that across this presentation, as well as some other competitors on the market. So we have something like coding packages like R and Python, as well as NanoString's Data Analysis Suite, or DSPDA. So you may have some experience with these items, but when it comes throughout all of these different steps that we've talked about through this masterclass, and even here with spatial differential expression and methods that you might choose for this, when you're considering this step, choosing a differential expression method and model is not often very intuitive, and it requires you to have a lot of statistical and spatial knowledge. And so if you don't have time for that or haven't had time to learn that, these current platforms, including coding packages and data analysis suite, the DSPDA, neither of these options allow you an intuitive or user-friendly or consistent option to select a differential expression model and understand and feel confident in that model. They do offer flexibility in choosing various models, and with coding packages, it is often transparent in what's being chosen, but you don't understand unless you've had the time to set aside learning and hours to become an expert in both spatial and statistical knowledge to understand what may best fit your model. Otherwise, this can be a very black void, and you may not understand the implications that can happen to your data. So the steps that Rosalind has taken, and what we're going to talk about today, is allowing an intuitive, user-friendly, and consistent, as well as flexible and transparent model to allow a very, again, flexible and transparent way to apply a spatial-oriented differential expression model applied to your data set that you can feel confident in every time you run a comparison. And so when we're talking about statistical tests and models, depending on your field, depending on your expertise, depending on your history, you probably think of 3 to 4 different types of stats tests and differential comparison tests right off the bat. There's a million of them that you can think about. I put a few examples here, and some of these are specific tests like Lemma or student t-test or Wilcoxon. And some of them are generic tests. So we have regression models, we have linear mixed models. So all of these different things, there's a ton of different models. And so when you actually think about it, how do you know what the right model is to actually compare your data and feel confident that the results you're seeing are correctly set up for what you want to visualize? So that's what we're going to talk about. So in general, what are some considerations you want to think about when looking at differential expression? Some items you want to consider are the technology of your data collection. What is the actual technology? Was it sequencing? Was it probe hybridization? Did you perform manual counting? Do you have something like qualitative data? There's a million different things that you could perform here with technology. And that is all going to play into what your data looks like, its noise, its background, its variance, all those things come into play. What about the biology of your data collection? Did you take technical replicates? How about biological? Do you have both? What's What's the difference? How about cells? Is your biology from cell lines? Did you have enough cell lines? Did you take it from real tissue? What is your genome that you're using? Do you have mouse line? Do you have human tissue? So all these things are important. Are you taking bulk data? Or what we're focusing on today, which is spatial. So all of these items have a direct effect on the data that you're actually expecting and how you want to actually analyze these results. And then your data set and model. Do you expect that the actual data that you have is representative of the population and model that you're trying to show? What does your data distribution look like? Do you have high variance? Do you have normal distribution? Do you know? What's your intended level of significance or measure of confidence that you place on this results? So is this a pilot study? Do you not have a high level of measurement? Is this a phase 4 clinical study and it's extremely large and a specific level? So all of these things have a different intent and a measurement that you have, and you have designed it that way, and you need to be aware of how you designed it. And then how do you intend to compare your data itself or these relationships? Are you looking at a generic difference between means? Are you looking at the same variables, like a pre and post study, multiple different groups, or a one sample versus one sample? So there's a million different things you could do here. And then of course, additional corrections to consider. So a lot of these things should already be corrected from if you ran normalization correctly, which is in our previous MasterClass session. And then there's other things that could still be needed to correct. So maybe technical or biological adjustments. So all of these different things can come into play that you may not realize is important to consider when you're running differential expression. So something that I really want to point out is that differential expression analysis is actually a very complex process. It involves many different steps, and it can be influenced by many factors, such as the ones I mentioned above, as well as the normalization factor that or normalization method as well. So many different things can affect this. So if you want to select the best method, you have to be aware of all of these factors that can affect your data and results, as well as the assumptions that are made within a selected differential expression model. So if you're not aware of these things, this can quickly become very complex, and you can easily select the incorrect method for your data. So if we start looking specifically at GeoMx WTA data for these different items that I highlighted, what is the technology for GeoMx WTA? This is actually a probe hybridization technology. It's similar to microarray. It does include sequencing as a secondary effect but it is a probe hybridization technology. So it has probe hybridization with secondary sequencing. So you're going to actually expect noise from both of those items. What does the biology look like? This is primarily spatial data. However, the next pieces are actually going to be down to your model and your design. Did you collect technical and biological replicates in your AOIs? Do you know what that means? Did you know how to take those? What about cells or tissue data? Did you take cell lines? Did you take tissue data? Did you take from mouse tissue, rat tissue, human tissue? All those things are going to have different variances. What about your data set size and model? Typically, after performing our second step, which is our AOI and gene removal, as well as normalizing your data, you'll feel pretty confident in thousands of genes, but this can vary depending, again, on your model. And then do you expect the data set size to represent the population that you're trying to mimic and model? And what level of significance do you expect to confidently represent this model, phase, and field? Are you doing a very quick, small pilot study? In that case, something like a p-value of 0.1 may be something to look at. You don't expect some strong significance if you're doing a very small discovery-based pilot study. Or is this a late phase clinical study where you need something like a p 0, 1. So you're looking for very high significance to release this into actual clinical drug release. So this can have a lot of different effects depending on where you're actually at in your studies and if you've performed something like a power analysis. And then do you expect additional corrections? So do you expect a high level of technical or biological variation factors to affect your background? Do you need to correct for these things, and do you expect them to not have been corrected by your normalization? So something we also want to remember is that the spatial field is fairly new. This is a 5 to 10 year field that just got, this is still fairly new. It's still being evolved. And because of that, optimal comparative methods are still evolving with it. So what we're going to talk about today is a lot of methods that are currently being used and what may currently be the gold standard, but I would expect these to continue to evolve as we learn more about best methods for spatial data. But to best consider this, I have a beautiful image of how you want to look at this statistical model. So the first thing to understand, which we just talked about, was the method, the technology, your model itself, what your data looks like. But then you also want to consider those next steps, which are those replicates and the comparisons that you want to set up. So when you're thinking about most common GeoMx model considerations and how you want to set these comparisons up, some of the most common ways to set these up are in these images here. Let me get my little red light. So you can see that the most common ways to set these up are within the same tissue. So you can see in this same tissue slide, you have a pink region and you have a green region. So maybe the pink is a tumor and the green is tumor microenvironment. So maybe you're comparing the tumor to the tumor microenvironment within the same tissue. That is a type of comparison that involves a specific intra-tissue comparison, versus where you may want to compare a healthy pink region to a diseased pink region across different tissues. Now you have an inter-tissue comparison. And it doesn't matter if there's multiple slides and different tissues involved, but as long as you're comparing within the same tissues, even if there's multiple, or across different tissues entirely. The point is how you're setting this comparison up. Are you comparing within that same type of tissue regions or not? So this is something unique to spatial that makes this really important to understand. Most importantly, because what you're going to expect is this point here, which is if you have some significant up or down regulations in your genes, you need to understand that there's assumptions in a lot of tests where you have independence versus non-independence. What that means is in spatial here, if we had 3 AOIs in this pink region, and let's say that that tumor-related genes are all upregulated in this pink region, which makes sense, it's going to say in most statistical tests, they assume independence, which means it's going to assume that all those AOIs taken from this pink region are assumed to be independent values, which means it actually thinks those are all unique samples taken from different patients or different tissues or different cell lines. So it thinks they're unique samples and not within the same tissue, which is this unique spatial model here. They're actually within the same unique tissue, and so they're actually non-independent. They are all dependent on each other here. They're non-independent. And so if we have upregulation or downregulation of these same genes within the same tissue, if they're assumed to be independent tissues, you're going to have higher significance in your genes. So maybe this tumor gene A, B, and C show a fold change of 20 because it thinks that they're all from different tissues, when instead it should recognize that they all come from the same tissue, and it shouldn't be as strong of an upregulation as what you're showing. And this can be really confusing to explain here, so we're going to talk about it through the next couple of slides. But that's why spatial is pretty complex, is because we're not typically taking from all independent tissues like we do for bulk bulk RNA-seq, for bulk single cell, or for cell tissue lines. And so this is where spatial starts to get complex. Additionally, there's another layer where we're often taking from the whole transcriptome. And so after removal of low expressing genes, you on average have 6,000 to 12,000 genes. However, those 6,000 to 12,000 genes are associated with one AOI. A lot of statistical tests also think that these genes are independent values. So you also have to make sure that those independent genes are not N equals 6 to 12,000, and it's associated with that N equals 1 AOI. So you can already start to see how the spatial can be really complex. And if you start to dissociate and make these all independent values, you can easily start to have misrepresented results and of all these AOIs. Let's say you had 80 AOIs. They're actually from 3 different tissues, but now your model looks like it has N equals 80 instead of N equals 3 different tissues. So that's how this starts to easily start to get out of hand and misrepresent the strength of your data. So some common models that are applied to GeoMx WTA data is student t-test, Wilcoxon t-test, lemma, linear mixed model, and linear mixed model with random slope. So we're going to talk about each of these models, the assumptions under these tests, and why they may or may not fit something like GeoMx WTA data. And then Rosalind specifically applies lemma, linear mixed model, and linear mixed model with random slope, and I'll discuss how we apply that and try to make this easier for you as the user. So the first ones I'm going to talk about is something that most people are familiar with, which is the student t-test. It's a beautiful test. It's the first thing you learn about. And when you're comparing data sets in your first stats test, and you're comparing, maybe some simple tests that you use in biology, maybe some qualitative data, maybe some cell line data, student t-tests are beautiful to use. However, this is not a great test to use for spatial GeoMx WTA data. The student t-test assumes that all data is normally distributed within equal variances. Totally fine. This is called a parametric test. It has a counterpart called Wilcoxon. So essentially, if you don't believe that you have enough N in your study to have normally distributed data, you're going to use the non-parametric counterpart to the student t-test called Wilcoxon. So this essentially says, I don't think that my data has enough strength to have normally distributed data. So if you have enough N to believe that your data is normally distributed, you'll use the student t-test. If you don't have enough N, you'll use Wilcoxon. The student t-test will compare means of 2 groups and determine if the single group differs from that known value using a standard deviation. This should sound familiar. The Wilcoxon will test the ordering of the data and creates ranks that can be considered a testing for shifts of the median values. So this makes it less sensitive to outliers. Because with Wilcoxon, when you're assuming that you have more variance in your data because you don't have standard distribution, you're going to assume you have more outliers because your data just looks that way. You don't have true outliers, you just have more variance in your data. So you want it to be less sensitive to your outliers. However, both of these tests assume independence in data. So that means that neither of these best fit for these spatial data sets that we discussed. So we're biological tissue where you have multiple AOIs taken from the same tissue, and you also have thousands of genes associated with one AOI and not one data point for one sample. These spatial data sets are actually non-independent and hierarchical data sets. So by assuming independence in their data sets, as well as typically assuming one data point is associated with one sample, these tests do not best fit spatial data and can greatly misrepresent your true results. So instead, let's talk about something like Lemma. This is a moderated t-test that was designed for RNA microarray datasets and other genomic and sequencing technologies. So consider something like very large gene pools. This is particularly useful if the mean of your features, so that's gene expression over probe signals. So again, GeoMx WTA is similar to a microarray. It's a probe hybridization technology. So it's particularly useful if you're expecting a large distribution range, and particularly with a high variance. So if you want to hear more about that, we talked a lot about the variance in this data in our last MasterClass series, talking about normalization selection. So this method utilizes an average variance based on all genes within a data set. It determines an individual sample's variance and adjusts the values towards an expected variance. So really fancy explanation, but essentially this works very, very well for large gene data sets. And it does not depend on the assumption of standard distribution. So first, it doesn't have an assumption of standard distribution, and it rather takes into account the mean over variance of different genes within the same sample's model fitting. So it essentially creates a model fitting according to the different genes that fit within that same sample. So it actually creates this hierarchical model saying we understand that there are thousands of genes that are not independent values. These thousands of genes actually associate and fit to this one sample. So we're already getting a lot better here. It says, okay, these 6,000 to 12,000 genes fit to this one AOI. These are not 6,000 to 12,000 independent values. So we're already getting a much better fit to what we want to see in our spatial data. But we also have a similar non-assumption of standard distribution. So this is similar to what we saw in Wilcoxon, where maybe this is where we would want something with less power, right? So this is something where you have less N in your data set. So maybe we can do something better. So now we have something called linear mixed model, or the LMM. This is a statistical model that analyzes non-independent, multi-level, hierarchical, or correlated data. So it already sounds pretty good, right? This is a type of regression model that accounts for both fixed and random effects. It has high flexibility to model means, variances, and covariances of data and fit multi-level, hierarchical linear, and random coefficient models. So that sounds pretty good, right? So let's talk about it. The linear mixed model allows the user to account for biological subsampling per tissue. Great. This is exactly what we need for GeoMx WTA data. So essentially, this allows us to create non-independence as well as a hierarchical structure. So that allows us to see we have non-independent AOIs, so multiple AOIs per tissue, as well as hierarchical structure, which is thousands of genes per AOI. This allows us to create statistical adjustments for multiple AOIs taken per tissue, allowing for the same, taking into account the same biological tissue entity and not as independent observations as the other statistical tests assume. So there are multiple different LMM models, and this is where we're going to kind of talk more into effect here, but this is the exact model that we currently want for how GeoMx WTA Spatial is designed. So the two main models that we're going to talk about is how we're setting up our comparison. So we talked that there's multiple models, this is really complex, but the main thing you want to consider is how you actually compare your data in GeoMx. So the first one is, are you comparing across different tissues? Even it can be multiple tissues on the same slide across many slides, but are you comparing that same region across different tissues? So in this example, right, we have a healthy tissues and we have diseased tissues, and we're comparing the tumor region across those different tissues. So in this case, this is an inter-tissue comparison. In that case, we are not introducing a random slope. If we're comparing within the same tissue itself, so tumor to tumor microenvironment, then we want to introduce a random slope. So what does this mean? And how can we figure this out? So Rosalind is going to actually do this for you. So this is really complex, right? Wouldn't it be nice if something could do this for you? So we are going to talk more in depth about how Rosalind can help you figure this out automatically. And so we'll talk a bit more about that, but essentially we will utilize something called the tissue ID attribute. The tissue ID refers to the origin tissue that the AOI was collected from, such as the patient, subject, animal, or biopsy. So Rosalind will apply the overlap of your tissue IDs in your comparison to determine the best model fit for you. So for example, if you had 5 AOIs that were collected from this same tissue, so let's say you took 5 AOIs, 2 were from the pink region, 3 were from the green, these would have the same tissue ID. So maybe it was like mouse 32, so they would have mouse 32 as their tissue ID. Or maybe it was patient 732, so then they would have patient 732 as their tissue ID. But if you have AOIs from different tissues, then their tissue IDs will not have that same identifier. For intertissue comparison, a majority of, so basically we're going to look at those tissue IDs and see how those overlap to determine how your comparison is set up. So you're going to set up that comparison and we're going to see if they overlap. If you set up an inter-tissue comparison, we're going to see that a majority of your tissue IDs do not overlap within that comparison. And this will tell us that the comparison takes place mostly across different biological tissues. And this will not require an adjustment for that biological subsampling that takes place within the same tissue. So essentially what you're going to get is this equation here. This is a linear mixed model. So we're going to have a test variable. So in this case, they're comparing across regions. So we're automatically going to apply this test variable. So if you wanted to compare against disease or date or, whatever your test variable is, we correct for that. And then your random intercept. So the random intercept that's going to be corrected for is your tissue ID, as we need to, the most variance that we can see in this model is that same tissue regions. The second example that can happen is a comparison setup within the same tissues. So you can again have multiple tissues, you can have multiple tissues across multiple slides, but what we would see is that a majority of those tissue IDs will overlap in this comparison that's set up. And so this will tell us that a majority of that comparison is taking place within the same biological tissues, and we need to make an additional adjustment to consider for that biological subsampling by introducing a random slope. So this is just an additional adjustment that helps us understand, for example, here we have individual tissues, right? So maybe this is mouse 1, 2, 3, and 4, 5, and 6. So these are different tissues. However, when you create a comparison within the same tissue, you need to make an additional adjustment so that the model understands that we're comparing regions within that same biological sample itself, so that these AOIs are again non-independent to another level that they're compared within that same sample. And so you want to add this random slope to additionally adjust for that. And again, that same random intercept will be here as well as the test variable. And so again, I hope this sounded really confusing, but that you understood it, because the most important thing in these is there are a lot of different statistical tests, but it's really important to understand this. But unless you have some understanding in statistics and spatial, it can be really confusing. So I hope this made sense to you, but the most important thing to remember is that you shouldn't have to have a degree in statistics to feel confident that your statistics are set up best for you in a software. And so Rosalind is setting this up for you so that you can feel confident in your results. And so, for example, what you'll see in Rosalind is you'll be able to set up a comparison for a GeoMx experiment. And again, we're going to use that tissue ID to determine the best fit model for you. So first, if we see that a majority of that tissue ID overlaps, we will set up a linear mixed model with that random slope adjustment, because we'll see that a majority takes place in that same tissue. So we'll want to correct for that. Well, if we see a majority of tissue ID does not overlap, then we'll see that they take place across mostly different tissues, and we will still set up linear mixed model, but without that random slope adjustment. However, there are some exceptions to these rules that, again, these are not beautiful 1-to-1, 2-to-2. There are a lot of different things that can come into play here and affect how this matters. And again, we take that into effect for you. So let's say you have this unique situation where all your tissue ID are actually unique. Maybe you did a microarray or biopsy punches where you actually only have very small sections and each of your tissue ID are unique. So you don't have any of that subsampling. This is actually so the reason we use tissue ID for the intercept is because you're going to have multiple AOIs taken per tissue. So that's best fit. However, if you have this unique situation, the best fit is actually adjusting your slope intercept to slide within your linear mixed model. So we're actually going to do that for you. Additionally, maybe you have a majority of tissue ID overlaps, and so we're going to run linear mixed model with random slope, but maybe you don't have enough power within that analysis to introduce that random slope correction. So a lot of these things can fall into effect. So we're going to automatically correct with a linear mixed model without that random slope adjustment, because this requires an additional fit. So not enough power, maybe a lack of enough AOIs, so small sample size. You could also have extreme outliers, so this can have a disproportionate effect on the estimation of your parameters, so like these random effects, or also unbalanced data. So this is something like an unequal group size across these levels of effects. So this can actually lead to difficulties in estimating random effects, and this can cause to issues in convergence and lead to a poor fit. So a lot of these things within GeoMx data can actually cause the model not to actually fit, and that will just let us, oops, I didn't mean to do that, but this will lead us to actually refit your model without random slope. The same situation can occur with linear mixed model without random slope. So if you still don't have enough power in this fit, we will then run lemma. So remember that lemma was a moderated t-test that was assuming non-normal distribution. So if we can't fit either of these models, we're going to run lemma with assumed non-normal distribution and high variance in your data, but still fits hierarchically with all your genes assumed to their each AOI. So there's still a best fit run model for what we can run. And the most important thing to remember is no matter what you use or where you're running your data, you need to remember to confirm the statistical test that you are running. If you don't know what statistical test is running your differential expression data, it's really important to confirm that test and also confirm that they are appropriate for your models and that your data is not violating the assumptions. And so with that, I'm going to quickly show you what this looks like in Rosalind and how you can set up a comparison and how we can confirm your data. And so in Rosalind, we're gonna start from where we left off in our last session, which was at our quantile normalized data. And so you can see the data, a lot of the graphs that we left off with. I'll quickly show you this MDS plot. And so this MDS plot is similar to a PCA plot, but it's on a two-dimensional space. A graph will be shown for every single attribute that is set up for your data. So I am going to show you this one. So for this data, it's going to show you the similarity of your different samples in space. And so for this data, these are different regions of the brain after stroke. And so what you can see here is that we actually have a high similarity between my core region. So this is where the stroke occurred. And then our different regions here. So we have the subcortex, so these high specific. So we have all goes here. We have specific cell types in each of these regions. And then we have a huge convergence of all these types after here. And so what you can't see, let's see if it's here. I don't think I have it on here, but these were actually taken at different time points. And so what we showed is that at 3 days post-stroke, we had a high differentiation in each of these regions, but at 7 days, there was less differentiation of the different regions. And so these are helpful in actually kind of quickly looking at your data and showing any similarity and confirming your models. So this is a really helpful graph to see. And then in our discovery and analysis, if this is your first time on Rosalind, I'm going to quickly show you how to set up a comparison and look at your data. So what we first do is we will confirm for you when you run a comparison what model was run. So I ran a few just so you can actually see. We will confirm for you if it was run as linear mixed model or lemma. And to create a comparison, it is extremely easy. So it is a drag and drop manner. You create these things called attributes. So these are any annotations that are important to you. So in this one, we created regions, conditions. So this is a stroke model where we gave a stroke, MCAO, and applied a treatment for neuregulin. And so you can create, you drag and drop them. So I'm going to drag and drop MCAO over here and neuregulin in my condition box. You can see that the box is automatically changed, as did my comparison name. And so just like that, it's set up. You can see how many samples are set up in each of these categories. You can edit the samples themselves. You can take one in and out. And we also have the select subset. So right now we actually have multiple different regions within these conditions. And so we'd actually want to further subset this data. So we can select region and then select which subset. So maybe I want to do core and save that. And now we have 8 in 6 samples here. So now we have the core of my MCAO and the core of my neuregulin treatment being compared. You can also drag and drop those yourself as well. So we can drag and drop core here and do that. And so this is an easy way to set up your data. And you can do this and compare different regions as well. So maybe we just want to compare. So this one, we're comparing the same condition in different regions. So this is probably going to do an overlap in the same tissues. So we have the same stroke tissues, but different regions. So this is going to be intra-tissue. And then for this one, we have the same region across different tissues. So this is going to be inter-tissue comparison. And so we can run each of those. And then you just hit save and run, and it will create the comparison, just like that. And we will run through that tree that I showed you, and it will essentially set up the best fit model for your data. And then once you have your data, you can actually click into them and select which one you prefer. If this is your first time on Rosalind, this may be new to you, so I'll give you a quick intro. So we have interactive data analysis. It will show you how many genes are upregulated or differentially expressed. We will apply an automatic fold change. So the default will be upregulated, downregulated by 1.5 with a p-adjust of 0.05. And you can see your upregulated genes in green and down in purple, right here. You can hover over them, and so you can see all this is changing. So you can change this, you can click on one, you can see how it looks. You can change this to a box plot and see how it's viewed. You can view a hierarchical heat map. You can see the volcano plot. You can click on each one and download them. We have pathways over here. So this is going to be an enrichment summary that we actually perform across all pathways. But we have over 50 different databases of pathways to choose from and explore and see where you see significance. If you'd like to change your filters, from the default, you can create a new filter. So I have a much more permissive filter at upregulated, downregulated 1.2 with a p-value of 0.05. And you can see that you can actually seamlessly switch between them and all your data will switch. And so this one has about 3,000 significantly changed genes. And so you can absolutely change these data points. And you can see with this data, this is a linear mixed model, and you're still going to get significantly changed genes depending on your model and your power of your data set. So if we go back here and we go to a similar one with lemma, we can't directly compare these results since lemma is going to be a much less powerful data set. But you can see that there's still a significant amount of genes. You can see some of these similar genes here. But since this was fitted a little bit differently, you're going to see a little bit of different results here. But yeah. So this one I set up to have a lower amount of n. So I removed some samples so that this would run on n of 2 versus 2. And so it would run at lemma versus these linear mixed results. But yeah, and so there's lots to explore in differential expression. You can share your data with collaborators, and by creating new filters, you can actually seamlessly switch between them. And so you don't have to worry about destroying your data if your p-values change, and you can explore your pathways in deeper exploration with our magnifying glass. And so there's lots that you can do. And with that, I'm going to go back to the PowerPoint, which is here, and finish this off. So this was the end of our last session. Thank you all for joining us. And that's it. I will take any questions.
Thank you, Jessica. That was a really informative and fascinating listen about all the different strategies for differential expression. Thank you for taking us through all the options and laying out the pros and cons. Hopefully that benefits everyone that's listening to this masterclass. So we do have a question that came in, and it seems like it's related both to lemma, so differential expression, and to normalization. And can you address maybe how, if you're seeing instability in the housekeeping genes, how is that related to, or does that impact normalization or the performing of lemma for differential expression?
If we're working with GeoMx WTA data, I wouldn't recommend looking at housekeeping genes. So housekeeping genes are almost negligible when we're looking at normalization. But I would say if you're interested in looking at them in general, with GeoMx, it's a little bit difficult because you most likely have very specialized areas that you're taking from. This is a probe hybridization. This is in situ. And so if, let's say you're collecting from targeted T cells, you're segmenting from T cells, and you're segmenting from a tumor, and you're segmenting from B cells. If you're doing something like that, I mean, housekeepers are no longer going to even apply. If you're taking from, let's say, just general AOIs, even if you're just taking whole AOI circles and they're the same region, they're the same circles sizes or diameters or whatever, and even that, if you're taking from, for my example, right, even mine were different areas of the brain, so they were like the subcortex and the corpus callosum and the area of necrosis in the brain. So even if I took from those, my housekeeping genes would be completely different because one area is necrotic, one area is has high oligodendrocytes, and it's an area with apoptosis, and they're just, everything's very volatile in the way that GeoMx is set up, that housekeeping genes have been shown to not be the best normalization method for GeoMx WTA for how it's working. So in how I would look at something like lemma, I would recommend, using a normalization method that doesn't rely on housekeeping genes for this. Housekeeping genes are best for smaller data sets, something like nCounter where you have 400 to 800 genes, and you expect something like bulk where most of your genes should show like a normal distribution. So I don't think I would apply those two together.
Thank you for that answer. That is all the questions we currently have. We'll give it a few more seconds here to see if anyone else has questions that they can ask Jessica.
Yeah. I think I see some questions. So if genes of interest are not present in filtered data, then what can we do about that? So Rosalind, if you missed this session on AOI and gene removal, Rosalind has a feature for genes of interest. So you create your own list of genes of interest and they will not be removed from your data set. So my answer would be do it in Rosalind. And then I think there was one more. Distance. Okay. Is there a method for including continuous distance measurement in your intra-tissue comparison? For example, tumor center to margin to healthy tissue. So I think my understanding of this question is, are we looking at, is there a way to look at distance like how you take AOI measurements and in distance, and maybe this is someone who also took, I forget the way that they can take this in GeoMx, but you can do concentric circles and take donut segments, and it's like 200 microns or whatnot, and you have these set distances. So I would say for this question, I don't believe that this is a differentially expressed variation that needs to be accounted for. But what I would say is if this is a variable that you want to compare, I would create an annotation or a attribute within Rosalind if you're setting out with Rosalind. And essentially you would call it maybe distance. And if you know their distances, you could have it so, maybe you call it that. Maybe you call it tumor center, or you call it margin or healthy tissue. That's almost how my stroke data was set up. So those were different distances from the stroke damage. And so they were just called those regions. But if the distance itself was important, then you could absolutely create that as a variable rather than the region itself. And you could compare those in its differential expression and see if you see any differences across your different AOIs. If they're taken, if you have multiple AOIs that have those distances regularly taken, then you have a model set up. However, if I wouldn't say it's something that needs to be corrected for, if that makes sense.
And Jessica, we just had another question come in. Can we select a list of differentially expressed genes or pathways of interest and generate bubble plots using fold change and p-value?
So you can select a list of DE genes and create a list. And by creating the list in Rosalind, you can visualize it in multiple different ways. At the moment, I don't think you can select pathways of interest for visualization. We are working on bubble plot visualizations. They're not there yet, but we have had a lot of interest in that, and so it is something that's like on our to-do list, but I couldn't give you a date for that.
And one other thing I'll add on to that, we're currently working on fully customizable figures directly in the platform. So we're starting with the volcano plot, MA plot on the differential expression page, also called interactive analysis, where you can fully customize the volcano plot change the color, the text size, the labels, cutoffs, export in a variety of formats such as PDF, PNG, EPS, SVG, and specify your resolution. So we're going to be adding this fully customization option to all of the figures within the Rosalind platform. It's going to be rolling out slowly, one at a time as we get them up and running. And bubble plots is on that roadmap as well. So that is a common request. So we will be addressing that one.
Yeah, great. Any last questions? Great. Thank you all for joining, and I hope this has been helpful. I think there was one more. Oh, yeah. Thank you all for joining. I hope this has been really helpful to you all, and these will all be recorded. This one should be up in a day or two. And I hope, this has been really helpful to your data analysis and what Rosalind can do for spatial. I know this is a new field, so it's, always something new to learn. So really excited to have been a part of this and helping share, some knowledge in spatial and how to approach it. And so really excited that you all have joined us for this session, and I hope to continue doing this and learn more. So thank you all.